share my screen. Okay, can everyone see my screen?
Yeah.
Yeah.
Okay. Hold on one second, sorry. Okay. Okay. Everyone's seeing my PowerPoint, right?
Yes.
Okay.
Yep.
Just want to make sure because sometimes it shows things, other things. Okay. So announcements wise, quiz number 9 was counted via attendance because I covered a lot of material in one go. So, so if you were here last week, you got points for attendance and the quiz. Okay. Quiz 10 will be out tonight. If you haven't uploaded presentations yet, please do so. If you don't see, if you've already presented and you don't see a grade yet, it's because you haven't uploaded your PowerPoint yet. So I just realized one thing, that next Wednesday is your exam. So one of the other things is that I have to go asynchronous with my 4:30 p.m. session. So since this 4:30 p.m. session is going asynchronous, I would like to do that with both of my other sections because for one thing, you guys have an exam right before our discussion section, so we'll be a little bit, I guess, in so we won't be able, we won't have anything much to cover at that point for the next exam. So what I'm planning on is that, so what I'm covering today is pretty much questions based on transcription and translation. So next week, I'm gonna go asynchronous with you guys as well. But I'm gonna put up some material that involves chapter 17, which is one that we won't be able to cover before your exam, and it's still on your exam. So I'm gonna have that uploaded this week along with quiz 11. I will send out an email reminding you guys, and then quiz 10 and 11 will be out this week, so that way you guys can work on it until we meet next time. So that will be on the 18th. Are we okay with that?
Yes, sounds good.
Yes.
How are we getting the attendance or participation points for next week? Is it just through the quiz?
So one of the things I do, so with the quizzes that I've been doing 2 points each, those are attendance and quiz grades as well. So this week will be, this next week's quiz will be that way. So if you do the quiz, automatically you guys get a point for attendance. And then the percentage of your, like that overall grade out of 2. So that if you get like 1 wrong, you get 75% of 1 point. So 0.75 for your quiz grade. Does that make sense?
Yeah.
Like if you get 100% on the quiz, you get 1 point for the quiz.
Yeah.
Okay. Any other questions, concerns, or anything else?
So the class is only out of 25 points. So if we do the next quiz, we have, if you've gotten everything else, you have a 25 out of 25. What does that mean for the next 2 classes and the next 2 quizzes?
Again, it is, so again, the top the top 10 quiz grades and attendance grades will be counted into your grade. So if you've done perfect 25 out of 25 already, you can either skip the next two, like 11 and 12, or do it and it won't, if you do less, it won't affect your grade. But again,
Okay.
these quizzes are pretty much trying to get you guys to think about the concepts. That way you guys won't fall behind. So I won't say not do them, but it's up to you guys. Are we okay?
Yeah.
So presentations. Who is okay. Who wants to go first? Oh wait, let me. Am I sharing? Okay.
Can go.
Okay. Are you able to share your screen?
Yes. Are you see my screen?
Yes.
Okay. Alright. So my topic was translation, question 1. So the question was label the spatial and polarity relationships among the following translational components: ribosome, small and large subunits, growing polypeptide chains, amino acid attached to tRNA, mRNA, codon, and anticodon. So that was the question. And this is the answer, the correct answer, the label for the diagram. So here we have the polypeptide chain. This is one amino acid. We have the mRNA down here. And all of this is the large ribosomal subunit. And down here we have the small ribosomal subunit, and which we have the codon and the anticodon. So look at background information. Translation is the decoding of messenger RNA and using that information to build a polypeptide. So here we have the polypeptide. So in an mRNA, the instruction for building polypeptide come in groups of 3 nucleotides called codon. In translation, the codons of an mRNA are read in order from the 5N to the 3N, which is here, by molecules called the tRNA. So each tRNA has a anticodon, a set of 3 nucleotides that binds to a matching mRNA codon through base pairing. So the other end of the tRNA carries the amino acid, that is specified by the codon. So the end result would be a polypeptide whose amino acid sequence mirrors the sequence of codons in the mRNA. So this will be changed to large ribosomal subunit, and that's reference. All right, then.
Good. Does that make sense to everyone?
Yeah, it was good.
Yeah.
Good. Okay. Who wants to go next?
Okay, how do you end this?
I can go.
Okay. All right. Thank you.
Yep. Ooh, colorful.
I've never done this before, so it's kind of difficult for me to work Zoom. You can see my screen?
Yep.
Okay. So I'm also in transcription. My question was to give a brief explanation of what occurs in A site and P site on the ribosome. So basically, a ribosome contains 3 sites for translation, which is the amino acyl site, okay, polypeptide, how do you say that word? Peptidyl.
Peptidyl, yep.
Peptidyl site and the exit site. The P site contains a peptidyl tRNA. The A site contains an amino acyl tRNA. So there's 3 phases for translation in which the P site and the A site both have things going on. So I wrote in my notes like other things that are occurring at the same time, kind of to give it background for what's actually happening and why things are happening in A site and P site. So initiation occurs, which blocks the A site with the initiation factor 1. It'll block that so that the tRNA can bind with the P site for initiation, and so that it can attach to the mRNA and the tRNA to attract the large subunit. When once that's all together, the small subunit and the large subunit are now together, and it can start elongation. So that in the amino acyl tRNA with the second corresponding codon to the mRNA will bind to the A site. So what I'm trying to say is, here you have UUC, you need the second corresponding codon with the amino active tRNA to bind here. So that's what's happening first. And then the ester bond between the tRNA and the P site will break so that it can associate with the what is it called? The terminal amino acid can bind with the initial amino acid in the A site, from P site to A site. And then you need a bunch of different types of elongation factors and a enzyme called ribozymes to complete the elongation process. So the third phase, whoops, I forgot to write. Oh wait, no, this is still part of the second phase. So in the end of the second phase was translocation, which is the creation of the polypeptide chain. So in the P site, the A site will make the tNRA officially uncharged when the P site will make the tNRA uncharged while it moves to the E site, which is exit. And that's a brief occurrence because then it'll just keep moving quite fast. The A site will bring a brand new charged peptide group, tRNA to its site. Phase 3 is the termination phase, and in the termination phase, a stop codon will be read after approximately 30 amino acids, codons. No, I don't know. 30 amino acids are built. What are these called? Peptide groups. Okay, thanks. Once 30 of those approximately, I think that's the number, are created, a stop codon will appear and it'll be read by the A site, but nothing will be able to translate in from the tRNA. So nothing will occur, and it'll move to the P site while what are these guys called? Forgot what they're called. Oh, release factors. Release factor 1 or 2 will bind to A, and that will make okay, so it'll release the polypeptide chain for the peptidyl tRNA. So that'll just release this guy here. And then the F3, so RF3 is the release factor 3, will release the tRNA from the P site, which is this thing here. And that'll officially disassociate the large subunit from the small subunit. And that's it.
Good.
Sorry, it was so horribly performed.
It was okay. But I liked how detailed it was. So does that make sense to everyone though?
I can't
Yes.
answer. Just kidding.
Yeah.
That's good. Okay. So next, Akasha, are you still presenting today? Akasha?
Sorry. Yeah, no, my computer broke and I just didn't. Yeah, no, I'll do it next week.
So next week is the week that we're going asynchronous. So it'll be the following week if you do.
Yeah, that's what I meant. Yeah,
Okay.
my bad.
Okie dokie. Just got to make sure that you guys actually do the presentation though, because you know, it's a required thing. Okay. Can everyone see my screen again?
Yeah.
Okay, good. Let's see if technology likes me today. So, as I said, we're going to cover some questions on transcription and translation. In the following coding strand of DNA, exons are colored in black and introns are colored in red. So, exon, intron, right? What is the sequence of the complementary pre-mRNA? So when you guys read these questions, be careful when you see the words coding strand or template strand, because there's a difference how you would do get the sequence of the pre-mRNA. So in this case, what do we know? We know that it is the coding strand. To get the pre-mRNA sequence of the coding strand, the only thing you need to do is change the thymines to uracils. Okay? So in this case, you have, this is our coding strand DNA sequence. With that, if you do transcription, you end up with this sequence here, which is which essentially is changing the T's to U's. So that's how you get this sequence here. Does that make sense?
Yeah.
Okay.
Mhm.
Now we have a so consider the following segment of DNA. Assume that the top strand is the template strand used by RNA polymerase. So essentially this question here is asking the mRNA sequence, right? So in this case, we know that the top strand here, if this wasn't given and you have the template strand, you have this strand here, you would find the complementary strand, which is this guy. So the template strand is always complementary to the coding strand. So you get that coding strand, right? So what do we know? We know that this top strand is the template strand. When a template strand is involved, you use the complementary strand to get the transcribed mRNA. So in this case, when you go through transcription, you find you change the U's, the T's to U's in the coding strand. So that's how you get this.
Yeah, wouldn't it be flipped the other way though? Because so like the TTG would be lined up with the 3' end.
So one of the things you have to remember is that so when you do this, it's directly out of this sequence, right? So when you do that, the 5' end stays the same as the coding strand.
Okay.
Because when you read it, you're still reading it from 5' to 3'. Does that make sense?
Yeah.
So what do you guys think about this question? Suppose that a portion of double strand, this thing is getting in the way. Go away. Okay. Suppose that a portion of a double-stranded DNA in the middle of a large gene is transcribed by an RNA polymerase. As the polymerase moves through the sequence of the 6 bases shown in the diagram below, what is the corresponding sequence of the bases in the RNA that is produced? So what do you guys think?
I think it's, wait, no, never mind.
It's okay. So where do we start? What do we know?
You start from the 5'.
Yes, we can start from. So essentially with these questions, we should know what is given to us, right? We have both strands given, coding and template. So in this case, we're gonna use the coding strand to solve this mystery of a sequence, right? So we choose the coding strand, and then you end up with the answer as UGAGCC, because you're only changing the T to a U. And again, 5' to 3'. That's why it's flipped. Again, when you read a sequence like this, it's always read from 5' to 3', unless it's specifically stated 3' to 5', okay? Does that make sense?
Actually, could you go over it again? Because it looks like the way that the DNA strand is, it looks like 5' starts at GG.
So this is the template strand. This is the coding strand.
Okay.
Up here is the coding strand, right?
Okay.
This sequence here is the coding strand. So we're ignoring this side here and be like, nope, we're not looking at it. Nope. And we're gonna be like, okay, so we're like coding strand. Okay, so we read RNA polymerase reads from 5' to 3', right? So when we do that, the coding strand is just gonna change the T to a U. So that's how you end up with this.
Okay, well, that's simple enough.
Wait, but so if you would code it, wouldn't the A correspond with the T though? Right, so wouldn't it be like TCU CGG?
Okay, so we have to remember we're talking about RNA, so we don't have any T's.
I meant ACU CGG. Because it goes with the coding strand.
So when you're doing it from the coding strand itself, the only thing that changes is the T's to a U, because that's what has the code. So the only thing you change in that part at that point is the T's to a U, because that's where it's coded. Whereas if you had the template strand alone, that's when you do using this sequence down here, which would make it UGA. Does that make sense?
Well, I'm just saying, like, it doesn't. I thought it had to be complementary to the coding strand.
No, it's
Because doesn't the
So it's pretty much the same sequence at that point. Except the only thing that changes is the T because there is no thymine in RNA.
I guess I'm just confused. I could have sworn that you made RNA by using the coding strand, and then it's the complementary base pairs to the coding strand.
That's if you're working with the template strand. Trust me, I had this confused for like years before I figured
So
this
you're
out.
just copying the coding strand and then replacing T's with U's.
When it's the coding strand. When you have the template strand, that's when you do the complementary.
Okay.
Yeah.
All right.
Does that make sense though?
Yeah.
I know it can be a little confusing. I had it confused for years before I actually realized it. So need to start you guys off right. Okay. Any questions before we move on? No? Okay. But yeah, always if there's no 5' to 3' written out, make sure you guys write it in 5' to 3', because if it's not written out, it'll be automatically read as 5' to 3'. So if you put CCGAU, AGU, it would be read as 5' to 3', and then you'll get marked wrong. So be careful with the directions. Okay. So the genetic code is blank, meaning that an amino acid may be encoded by more than one codon. So what feature of the genetic code does that entail? Do you guys remember? I know lecture covered an entire slide of it, so.
Would it be A?
Nope,
Is it D?
nope,
Is it B?
B, yes. Okay, so the features of the genetic code. So when you look at the genetic code, it is degenerate. So degenerate itself means that in this case, for the genetic code, it means that more than one triplet codon can specify for one amino acid, right? Unambiguous means that each triplet can only specify one amino acid. So you can have more than one codon specifying for an amino acid, but each triplet can only pair with one amino acid. Does that make sense?
Yes.
Okay. Now we have something called commaless. This essentially means that if translation occurs, then you end up with codons being read without a break. Because like even if you hit a stop codon and you have codons after it, it won't read anything past that stop codon. And then you have non-overlapping. So any single ribonucleotide within mRNA is part of only one triplet. So ACA, UGA here, this A is part of this codon alone, and this U is part of this codon alone. It's not overlapping between, this A cannot be part of this guy. This cannot be a part of this guy, that kind of thing. Does that make sense? Yes, no, maybe so.
Yeah.
Okay. Then we have like, so next thing is it is nearly universal. So essentially this coding system, like the genetic code system, can be used within viruses, prokaryotes, archaea, and eukaryotes, right? Maybe some of them are missing a couple of them, but essentially it's nearly universal, as in it is conserved between different systems, right? Does that make sense?
Yes.
So that is why the answer to this question is degenerate, because it's essentially a term for redundancy in our genetic code. So we have 61 codons that code for 20 amino acids. So many of the amino acids end up having more than one codon that comes with it. Okay, so the answer to that is B. So this question, the sigma subunit of bacterial RNA polymerase is, what does it do? What do you guys think? Okay, I get, oh, there's some things in the chat. Let's see. Okay, it is not C. So where does sigma come in? Do you guys remember? We know it's prokaryotic.
I think it's like, oh, wait, no, never mind. I don't know that.
Is
We
it
know,
E?
oh. Nope. So RNA polymerase, guys, RNA polymerase should be screaming transcription. Binding to the ribosome occurs with translation.
Is it D?
Nope. So where is RNA polymerase going on? No? Okay. So this is like the first figure that we have, right? So we have the core enzyme, right? So this core enzyme is actually binding to the gene's promoter, right? So we have the alpha subunits, the beta prime, all come together with the omega. And it's like, okay, so sigma then comes in to the promoter and it helps the polymerase to the center. That way, once the, so this sigma here binds to that promoter area, this area of the promoter, and then ends up putting the catalytic center. So that center that the enzyme is, so that catalytic area and the sigma subunit comes together and it starts, it brings it to the initiation site. Does that make sense?
Yeah.
So that's essentially what the sigma subunit is up to. So in this interpretive drawing of heteroduplex formed between mRNA and its coding DNA, which is in pink, and what do the loops labeled A through G represent? So we have a mature RNA, and then we have a strand of DNA, the coding side of the DNA. And you have that, okay, it's like matching up here, right? So we have that. what are these loops? A, B, C, D, E, F, and G. What do you guys think that is? So what do we not have in mature RNAs?
Is the answer introns?
Yes. So essentially, mature RNAs do not have introns because they're spliced out and the exons are fused together. So when the DNA comes in, tries to match up with the mRNA, it pushes out the introns. That's why it ends up looped out. Does that make sense?
Yes.
The poly A tail of mRNAs does what? Or what's happening with that? Anybody has an idea?
Would it be 5? All of the above?
Yes. So that is correct. Choice 5. So so with transcription in eukaryotic mRNAs, they end up getting a poly A tail, so a string of adenine residues added on to the end, the 3' end. And the main function of this tail is to stabilize the mRNAs against degradation by the exonucleases. Does that make sense? So essentially that's what it is and what it does. Okay. So that is the answer for that. Okay, so what is true about tRNAs here?
Is it 5?
tRNAs do not have 2 subunits.
Four.
tRNAs can only hold one type of amino acid.
Is it 1?
Yes. Okay. So essentially, with these guys, the tRNAs can, so it's only one molecule, but it can come and bind with these areas like this. So that's how it comes with a secondary structure, right? Does that make sense? That's why it has double-stranded regions. Are we cool with tRNAs? Or do we hate them?
I kind of hate them.
Boo, right? Being all complicated for no reason.
I just feel like there's so much in these units. There's so many
I
things to know.
Yeah, it's quite a bit. Okay. An amino acyl tRNA synthase for valine is mutated so that it now attaches the amino acid glycine to the tRNA valine instead of valine. What will happen at translation? So choice A has there will be glycines at all valine positions and valines at all glycine positions. There will choice B says there will be valine at all glycine positions and glycines at all valine positions. Choice C is there will be valines at all glycine positions and at all valine positions. D is there will be glycines at all glycine positions and at all valine positions. And E is there will be no translation. What do you guys think?
D.
Yep. You guys are awesome at this. Okay, so essentially, because this tRNA valine is still recognizing the valine codon, but so the tRNA has the codon that codes for valine, like it has the codon and the anticodon, right? So the tRNA is essentially attaching whatever amino acid it has attached to. So in this case, due to this mutation, this tRNA valine is actually charged with glycine, right? So the glycine ends up inserted instead of valine. But so that gets happening with that. And then the glycine codons end up getting recognized by their own tRNA glycine. So the anticodon for the glycine codons will bring their normal glycine amino acids and they will be placed in their normal positions. So that is why it's D. Does that make sense?
Yeah.
During bacterial translation initiation, the mRNA does what? Anybody remembers? No? Maybe? So what words scream initiation and bacterial translation? What is the first thing that actually happens, actually?
Is it D?
Yep. Yep. So essentially the first thing that the mRNA does is it binds to the Shine-Dalgarno sequence and ends up to that 16S rRNA of the small ribosomal subunit. This binding ends up orienting this towards the start codon in the P site. That's when the FMET tRNA comes in and binds to that AUG codon, and the large subunit comes to be attached, which requires some GTP hydrolysis. So that is why the answer to this is D. Does that make sense?
Yeah.
During elongation phase of translation, the charged tRNA center enters the ribosome at the which site?
Is it the A site?
Yes, it is the A site. So guys, so remember the first thing they do is come in. When they enter, it's at the A site and then it gets shifted to the P site where you have a growing peptide. go on, right? And then the E site releases them, okay? So the answer to this is the A site. What do you guys know about peptidyl transferase? What do you guys think?
I believe it's C or D. Catalyzes the reaction that joins TNR. Yeah, maybe it's that one.
I wish you stuck with C. It's C is the answer. So essentially, peptidyl transferase is a ribozyme. So it's essential. So a ribozyme is essentially an RNA molecule that has catalytic activity. In this case, with bacterial ribosomes, this function is assigned to this specific ribosomal RNA, and it catalyzes the formation of peptide bonds between amino acids during elongation of translation, okay? So essentially with this guy, you have the amino acids going on, and it's like, so peptidyl transferase, always think peptide bond formation, okay? So it's allowing the formation of these peptide bonds to form. Does that make sense?
Could you just go through that one more time? Sorry.
Okay, so peptidyl transferase is essentially a ribozyme. So a ribozyme is a RNA molecule with catalytic activity. So it's like giving a boost to like a function to occur, right? So in this case, with bacterial ribosomes, this function is actually assigned to this specific ribosomal RNA, and it ends up catalyzing the formation of peptide bonds. So you know how we have to make peptide bonds at the P site, right? So when they're adding on each of the amino acids, this peptidyl transferase is allowing it to occur, allowing peptide bonds to form. Does that make sense?
Yes, thank you.
Okay.
Yeah.
In this drawing of hemoglobin, what levels of structures are visible? What do you guys think?
I think it's I'm pretty sure it's quaternary.
Okay, that is actually one of them.
So is it E?
So essentially, like, the only thing you cannot see in this guy is actually the primary structure, which is the sequence of amino acids themselves, right? So you have some folds here that end up having the secondary subunit structures in the backbone itself. Then you have the tertiary structures, which are like the alpha and beta chains and all that. And then interactions of all of these molecules, like the heme group and all of this different components, that makes up the quaternary structure. Does that make sense? Maybe.
Yeah, it does.
Okay. if there were 6 bases in DNA and still only 20 amino acids, how long would a codon need to be in order to specify an amino acid? So I know we haven't really gone over this one, so I'm gonna walk you through this guy. So in this case, you would start off with, so in this case, we're trying to look at how many combinations of codons can we have that gives you a minimum of 21 codons, right? Because you have at least 20 amino acids and then you have at least one stop codon, right? So in this case, if you have this guy, okay, so you have the different combinations of codons being equal to the number of bases you have raised to the power of the length of the codon. Since we don't know the number of different combinations that they are telling us, we're going to assume that each of these are a possible choice, but you're looking for the minimum number. So in this case, when you use 6 bases in a 2-base codon, that gives you 6 to the power of 2, which gives you 36 different combinations. That right there would be enough to encode all of the 20 amino acids and the stop codons, right? Because you have at least 20 codons to code for this guy. And then you have a couple of stop codons. Again, the code itself could be degenerate, so multiple of these codons could apply to the amino acids themselves. Does that make sense?
Yeah.
Okay. So this question, the wording is a little bit confusing here, but with nonsense suppressor mutations, so a nonsense mutation is essentially changing an amino acid to a stop codon, right? But when you have a suppressor mutation in a nonsense mutation, you end up with a so essentially this guy, the suppressor mutation, suppresses the effect of this guy. So it essentially would end up restoring the functionality of the nonsense mutation by allowing an amino acid to be added to the polypeptide chain. Does that make sense?
Yes.
Okay. So that is all I have for today. So I'm gonna put up like a study guide and a video for chapter 17 stuff. It might be more detailed than you guys need to learn. So make sure you guys look at your lecture material and make sure like you're looking at the right amount, okay? Because I know we didn't get to cover chapter 17 because for one thing, you guys only started translation in class. So, oh boy, this sounds wonderful. But any questions, concerns?